So our immunity will help us fight against the communicable disease. As you know that there are two types of disease which we come across. One that are communicable, which can be transmitted from one person to another person, while others are non-communicable, that cannot be transferred from one person to another person. The non-communicable diseases are, for example, diabetes, high blood pressure, then hypertension, migraine, etc. Merely coming in contact with the person who is diabetic or who is having hypertension, you cannot acquire that disease, and those diseases are called non-communicable disease. So in that, immunity plays no role. While those diseases which can be spread from one person to another person, they are called communicable disease or infectious disease. For an example, HIV, cough, flu, skin infections, etc. All can be spread from one person to another person, and they are called communicable disease, which we can communicate from one person to another person. Okay, so which are different modes of the transfer of infection? Can anyone recall any mode of transfer of infection? Anyone? How infection can spread from one person to another person? Yes?
Droplets cough.
So there are different means by which the infection can spread. First is the airborne infection. Then droplet infection. Then through the infected articles. Yes, please. Then through blood, through vectors. So these are few modes of transfer of infection from one person to another person. Airborne infection, that is the infection is spread through air. We are all aware about the COVID. So coronavirus was spreading through air. There are number of viral infections like flu, common cold. They all spread through air. So the organisms which can travel freely through air, they are called airborne infection. TB is one of them. Okay. Next is droplet infection. There are few infections which can be spread only when we come in contact with the droplets which are infected by the organisms, like coughing and sneezing. When we cough, when we sneeze, we throw out number of droplets, and those droplets carry some organisms, so they spread in the air. Now, the major benefit, or we can say a good side of the droplet infection is we can only get infection if we come in contact with those droplets. So the effect is transient. If some person is coughing or sneezing in the vicinity of your when you are present, then only you will get infection. You will think someone has coughed over here before an hour and you will get infection. Because the droplets are having their own weight, and that's why they will sediment, they will settle down after some time. That's why you won't get infection once if you visit the area which is infected or contaminated by sneezing or coughing after an hour or later. While in case of airborne infection, the infection can spread even after a long time because the microorganisms remain suspended in air for a long time if they are free, and that's why this can be consistent for a long time. This is a short time infection, that is, you can get infection only if you come in contact very recently. Then through infected articles, there are a number of articles. For an example, syringes, needles, blankets, towel, clothes, then okay, n number of articles which are sometimes responsible for the spread of the disease. We are well aware that HIV that spreads through blood or contaminated syringe or needles. The skin infections, majority of the fungal infections, they spread through the articles which are used by another person. like using the clothes or towels or beds of infected person. Okay, so they are spreading the infection. Then next is blood. By blood transfusion also there are a number of diseases which are transferred. Nearly all the diseases which are infecting the blood can be transferred through blood. So before blood transfusion we are testing the blood for presence of any infectious organism. Then vectors. Some diseases are carried by some organisms. Most common vectors are mosquitoes, flies, flea, etc. Okay, we all know that malaria, dengue, they are spread by mosquitoes. Then typhoid, that is spread by the flies, house fly, and flea. One of the example of the disease which is spread by flea is plague. Now, in case of plague, there are two vectors. This flea is a primary vector, which collects blood from an infected person. But the flea cannot travel from one place to another place easily, and that's why it uses a secondary vector. So, for plague, rats are secondary vector. They carry the flea from one place to another place. That flea transfer the infected blood to another person, and thereby that person gets infected. So in such case, we are having two vectors. One that carries the blood, another that carries the vector. So rats are secondary vectors for that. Clear? So these are different modes through which the infection can spread from one person to another person. Now, what are the factors which decide the severity of the infection or the chances of occurrence of a disease? So first, let us discuss the factors affecting infection. I'm going a little fast. If you have any question, you can ask, because we need to complete the chapter in a very short time. So I'm moving fast. So factors affecting infection. First of the factor is virulence. Virulence is the ability of the microorganism to invade. The body establish itself and cause the disease. So, first thing required for infection is the ability to invade. That ability to invade is known as invasiveness. And second thing which affects the disease is the establishment or the toxigenicity. Microorganisms, when invade the body, they will establish themselves, they will form the colonies. Once they multiply, they use the nutrients of normal cell. Now, the disease which are caused by organisms can be of two types. One, that are harming or affecting the body because of the toxins which they produce. And second is merely by their multiplication. For an example, throat infection. So in throat infection, what happens is, our throat is infected by microorganisms. The bacteria which are infecting the throat will consume the nutrients of normal cell. That will lead to death of the normal cells, and thereby we feel scratchy or itchy or coughing. While in some disease like tetanus, the microorganisms invade the body and they produce toxins, and those toxins are harmful to our body. Like as Clostridium tetani, it produces tetanus toxin. This tetanus toxin, in Gujarati we know, we call Dhanur. That is harmful. That's a neurotoxin, and it affects the body, and thereby it causes the harmful effect. So here, all the organisms are not capable of producing such toxins, and that's why the second thing which comes in the action is the toxigenicity. that is the ability to produce toxins. Now, all organisms have different invasiveness, that is the ability to invade the body. At the same time, they are having different toxigenicity as well, that is the ability to produce the toxins. For an example, the organisms which are responsible for common cold, the virus normally. Okay, common cold is normally caused by virus. So the virus of common cold can enter the body easily through respiratory tract. But when they enter, they are not producing any toxins. They are not producing harmful effects, much harmful effects. So they are having good invasiveness, but lesser toxigenicity. Their ability to produce toxin is less. At the same time, Clostridium tetani, it cannot invade the body if there is no broken skin. It can only enter the body through broken skin. It can only invade the body through broken skin, and this is the reason why after getting some cut from glass or rusted metal or anything, we go for the tetanus vaccine. બરોબર આપણે નોર્મલી કે કાટ લાગે કઈ લાગ્યું છે ખીલી લાગી છે એટલે પહેલા શું કે ધનુનું ઇન્જેક્શન લઈ લઈએ. That is because the Clostridium tetani can enter the body only through a open wound, that is a cut skin. Unless the skin is cut, it can never invade the body. It cannot enter the body. So it is having poor invasiveness. But once it enters the body, it produces a very harmful neurotoxin, that is tetanus toxin, and that is why it is having higher toxigenicity. Lower invasiveness, higher toxigenicity. Clear? Now, the toxins which are produced by bacteria are of two types. Broadly, they are categorized in two categories: endotoxins and exotoxins. Endotoxins are not water soluble. And they are only liberated or released when cell lysis occurs. So once the cell dies, they are liberated. While exotoxins are the metabolic products of bacteria. Actively multiplying bacteria. They are water soluble. And they can diffuse out to surrounding environment. So when the bacteria are growing, when they are multiplying, they can easily diffuse out of the bacteria to the surrounding environment, and thereby they can spread while the bacteria is still alive, still multiplying. You all are aware that in case of parenteral products, we are performing a test that is called pyrogen test or bacterial endotoxin test. Pyrogen test or bacterial endotoxin test, in which we are testing for the presence of pyrogens or bacterial endotoxins. They are the products of their lysis, that is normally they are constituents of their cell wall, while exotoxins are metabolic byproducts of the actively growing bacteria, and when the bacteria are alive, they are producing this toxin in high concentration, high amount, and that they are more harmful as compared to the endotoxins. The endotoxins are the complex of sugars, peptides and lipids, because they are the components of cell wall. And the natural cell wall is made up of the peptides, sugars and lipids. So they are the complex of sugars, peptides and lipids, while the exotoxins are the enzymes or polypeptides only. As These exotoxins are polypeptides of enzymes. They are thermolipid. They can be destroyed by heating up to 60 degrees centigrade temperature. If we heat a solution or material above 60 degrees centigrade, the exotoxins can be easily destroyed. And that is why, while manufacturing parenterals, we are never worried about the exotoxins. We are only worried about endotoxins. In all the books or literature pertaining to parenterals, you might have read that we perform the bacterial endotoxin test or pathogen test. We are never worried about the exotoxins because they are highly thermolipid. They can be easily destroyed. And during the sterilization by either autoclave or whatever one, we can easily destroy them. But endotoxins are thermostable. They cannot be destroyed easily. And for destroying the endotoxins, we need to heat at temperature above 160 degrees centigrade. And that's why they are cause of concern number of times. These endotoxins are mainly produced by gram-negative bacteria. Okay, all organisms are having their cell wall, but the major concern is with the gram-negative bacteria, while exotoxins are produced by mainly gram-positive bacteria. The endotoxins are less harmful. They only increase the body temperature. They don't do anything else. Okay. Less toxic or less harmful. They only increase the body temperature, and that's why they are called pyrogens. They increase the temperature, and that's why they are called pyrogens. So they will only increase the body temperature. There are no any other effects. While exotoxins are more toxic. Let me give an example. The botulinum toxin. Which is produced by Clostridium botulinum. 1 milligram of this toxin can kill 1 million mice. 1 milligram of this toxin can kill 1 million mice. So that is the ability of this toxin. In humans also, it is a neurotoxin, and that's why it can kill the person. While no endotoxin is having that ability. They will never kill. Okay. So the endotoxins, as I said, they are produced by the gram-negative microorganisms. These are produced by gram-positive, like Clostridium botulinum, Clostridium tetani, etc. While these are produced by the organisms like Staphylococcus aureus, Streptococcus, Mycobacterium, tuberculosis, etc. Okay. Any question in this, anyone? Getting it? So this is the first factor which affects the infection. Let us discuss the second factor. The second factor which affects the infection is the number of organisms. In what quantity we are getting infected by the microorganisms. And it is self-explanatory. If the number of organisms present or which are entering the body is high, there are more chances of infection. Now let me give a simple example. The food every day which we consume is definitely 110% infected by the microorganisms. There is practically no method of cooking which can kill all the microorganisms, or even though it is killed, the food will get re-infected because it's a rich source of nutrients. So if we keep the food open for some time, then also the microorganisms can re-enter. If we consume the food which is infected, then also we are not getting infected. We are not getting any disease. But if we consume a food which is infected highly with the microorganisms, that is, we keep the food for 2 days, and after 2 days if you consume the same food, you will get disease. Okay, normally we call it food poisoning because the number of organisms on that food is higher than the freshly prepared food, and this is the reason why we get infected. So number of organisms which are entering the body play a crucial role. It is the capacity of our body to neutralize, to kill small number of organisms, and that is why we don't get infected daily. Otherwise, you will get infection daily. We are daily coming across number of organisms through different routes, through water, through food, through air, through physical contact, everything. But still, we don't get infection daily because our body can handle less number of organisms. If the number of organisms is more, then only we will get infected. Here, so second factor as we discussed, number of organisms, and it is second factor. Third is the route of entry. For some organisms, the route of entry is also crucial. For an example, Neisseria gonorrhea. Neisseria gonorrhea, that is responsible for a sexually transmitted disease called gonorrhea. It only infects the person if these organisms enter through urinogenital tract. It only infects a person if it enters through urinogenital tract. Any other route will not affect. Okay? Then Klebsiella pneumoniae. That causes pneumonia. It only affects the person if these organisms enter through respiratory tract. Through any other route, it won't affect. Okay? Salmonella typhi. It will only affect if this is through respiratory. Salmonella typhi. It only infects the person if enters through GI. Typhoid. It is only affecting GI. Clear? So, route of entry is important. If organisms enter through any other route, they won't be able to produce the disease. So, because of their normal growth requirements, or because of the protective mechanisms present in that area. Though there are some exceptions over here as well. If there are some organisms like Clostridium or Mycobacterium. It can affect the body in multiple ways. If the Mycobacterium enters through respiratory tract, it can produce the pulmonary tuberculosis. Which is most common form of tuberculosis, which is affecting the lungs. So it can produce pulmonary tuberculosis. If it enters through GIT, it produces the intestinal tuberculosis. Very rare, but still there are occurrences of intestinal tuberculosis. If it can cross the GIT areas and it can pass to blood, or if it is injected through the veins by contaminated particles, then it can also infect the bones and can produce the bone tuberculosis if enters the blood. The entry may be through the ulcers as well. Sometimes when the people are having GI ulcers, they are more prone to some other disease as well, because of the ulceration, the protective barrier of this GIT is broken, and through that the microorganisms can easily invade. So they are more prone to a number of other diseases as well. Okay. So this is the organism which can cause the disease through multiple entry. Rest are having the specific entry mechanisms, and they can produce disease only if they enter through that particular route. Fourth mechanism of factor which affects the infection is the host defense. the person himself is able to resist the infection, then there are less chances of infection. Now, this host defense is working in three different layers. First is the prevention of entry. That is called first line of defense. Nature has provided us with number of mechanisms to protect ourselves from the infections. The largest barrier to entry is our skin. No organism can enter through the intact skin. Unless the skin is broken, no organism can invade, no organism can enter. And that is the largest barrier for the entry of the organisms for infection, which our body is endowed with. Second is the secretions. For an example, we are having different secretions. If organisms try to enter through oral route, first barrier which it comes across is the enzymes of the oral cavity, that is salivary enzymes. The salivary enzymes are capable of digesting the polysaccharides. In schools, you might have studied the reason. ગુજરાતમાં રોટલી ને ચાવવાથી રોટલી મીઠી લાગે છે. બરોબર? The reason is that starch which is present in wheat flour that is digested by the salivary enzymes and is broken down to the monosaccharides, and that's why the taste becomes sweeter. So microbial cell wall also contains, as I said, sugars, that is polysaccharides, and those polysaccharides are digested by the salivary enzymes, and hence some organisms are destroyed in the oral cavity itself. If they can enter further, they can pass further through the oral route to GIT. In our stomach, we are having proteolytic enzymes. Pepsin is capable of digesting proteins, second constituent of the cell wall of microorganisms. So, highly acidic pH, that is very low pH, and the enzymes, proteolytic enzymes, they will digest the cell wall. They will kill the microorganisms. If they can pass further from stomach to intestine, then in intestine we are having the lipolytic enzymes. They can digest lipids, the third constituent of the bacterial cell wall. As the bacterial cell wall is composed of sugars, proteins, and lipids. So now we are targeting the third component of the bacterial cell wall, that is lipids. They are digested in intestine. So that also we try to prevent the entry. Here there are three layers present in GIT: saliva, stomach, and lipolytic enzymes, which are present in the intestine. They will destroy the microorganisms. If they survive, then only they can enter the systemic circulation. That is the if they enter through eye, we are having lacrimal secretions. Lacrimal fluid. This lacrimal fluid is highly hypertonic. So, the ears are having higher concentration of salt. They are hypertonic in nature. Because of the hypertonicity, microorganisms cannot survive. At the same time, they are having some enzymes as well, which are capable of digesting the cell wall of microorganisms and they protect them. If they try to enter through nasal route, first thing, we are having the nasal hairs, and they are always moist. Okay, our body keeps it moist. So any dust particles or organisms, if they enter, they will be trapped in the nasal gas. If they travel further, they need to travel through a route which is not straight. બરાબર આપણે જો શ્વાસ લઈએ છીએ ને તો ઇનિશિયલી ઉપર જાય છે પછી નીચે આવે છે ડાયરેક્ટ શ્વાસ નથી જતો બરાબર now the mucosa of this region is also having constant secretion of highly viscous fluids, and those viscous fluids can trap the microorganisms. When any particles or any microorganism travels through any route, it cannot change its direction as quickly as that of the air. When we are breathing in, the air travels through the nasal route. Now, air changes its route easily, its direction easily, but the particles cannot, and that's why they will experience a centrifugal force. Because of that centrifugal force, they will be thrown away, and they will stick, they will adhere to this mucosa, which is having a very viscous secretion. They will track them. It will prevent the further transit of the organisms and it will provide the protection. Next is through the genital tract also. We are having number of secretions, and that secretions as well as the pH. The pH of the genital tract is higher than the normal pH. It is alkaline in nature, and that can also kill the microorganisms. It's also preventing the microbial entry. So we are provided with mechanisms which will prevent the microbial entry through all the routes. That is the first line of defense. Now, if organism enters the body after crossing all these barriers, then our second line of defense comes into action. That is called non-specific defense. This is second line of defense. You might have read your blood reports or blood reports from any of your relatives. When they get infected, there are some steps, some numbers in their blood count changes. Most common of them is the WBC. There is huge increase in the WBC if they are having any bacterial infection. Particularly in bacterial infection, WBC increases. In viral infection, CRP increases. C-rate protein increases. Okay, so all these are the parts of second line defense. Our blood is having number of such cells which are capable of killing the bacteria or virus. For example, phagocytes, natural killer cells, or NK cells. Then defenses. These are proteins. Then complement proteins. And macrophages. And many more. They will kill the antigen. Now we will use the word antigen. Anything which enters from outside and which can trigger the antibody production is called antigen. So when the antigen invades the body, all this will try to kill it non-specifically. Mind you, non-specifically. That is without even identifying it. For any infection, you may observe that your WBC, CRP, or any other indicators have increased. One of the common diagnostic parameter which doctors use is the NL ratio, neutrophil to lymphocyte ratio. That also changes when you get infected. So our body itself multiplies or increases the production of some specific type of cells which will help in defense, and thereby they will kill or try to utilize the antigen without even identifying that. So that is the part of second line defense. Let's discuss the phagocytosis. It's a normal thing. Very frequent question in the exams. So, the phagocytes are nothing but a type of WBC. So, what they do is, first step is the binding of antigen with the phagocytes. This binding is facilitated by other cells of our body as well, like complement proteins, opsonins, and the antibodies or immunoglobulins. They will facilitate the binding or adhesion of the antigen with the phagocyte. They will make or mark the antigen identifiable by the phagocytes, or they will trigger the attraction of phagocytes towards those antigens. Normally, in case of the immunoglobulins, the lower part of the immunoglobulins, as you know, the structure of immunoglobulin is like a gamma, or like this. Okay. So this part acts as a flag. When it binds with an antigen, it is only neutralizing or precipitating the antigen. It is not killing the antigen. But this part will act as a marker, as a flag for identification. And that's why the phagocytes can easily identify them. The function of the antibodies is only to make the antigen immobile or insoluble, or separate it from the bloodstream. That is, make it inactive. It will never kill it. It will never destroy it. The killing is facilitated by the phagocytes. So they will only make this cell identifiable by the phagocytes, and that's why the phagocytes are targeted to that cell. Then it binds with the phagocyte. The phagocytes then will engulf the cell. That is called endocytosis. And they will form a vacuum. In that vacuum, that is called phagosome. Now, in phagocytes, they are having the lysosome. That is the enzymes which can cause the lysis. They are called lysosomes. In the phagocyte, these lysosomes and these phagosomes will fuse together to form a phagolysosome. This lysosomal enzymes will cause the lysis, that is the breakdown of these cells, and they will totally neutralize or break down the antigen, and then they are thrown out. That is called exocytosis. So this is the entire cycle of the killing of the microorganisms, that is antigens, by the phagocytes. The entire process is known as phagocytosis. Phagocytosis, starting from binding, fusion, and exocytosis. Clear? And that is facilitated by normally the complement proteins, opsonins, and immunoglobulins. They make it identified. In case of bacterial disease. Now, in case of viral disease, we cannot go with this process. So, in case of viral disease, there are two mechanisms. One is the interferons. Normally, once a cell gets infected by virus, as you know that viruses are not having their own metabolic process. They will only ingest their RNA or DNA into the host cell. The host cell will multiply their RNA or DNA. And in such cases, we cannot isolate or differentiate the viral cells from the normal cells. It becomes difficult for them. And that is why the cells who are infected will produce interferons, INFs. These interferons will be liberated by these cells to the surrounding environment. So the surrounding cells can easily recognize easily identify that this cell has now been infected by virus. So those cells will produce the proteins which will provide defense against the virus, and that can block the replication of that viral DNA or RNA, that is viral genome in these surrounding cells. So that is one mechanism. And second is the natural killer cells or NK cells. They are called natural killers because they are part of the apoptosis mechanism. Apoptosis is a programmed cell death for preventing the cancers or preventing the unrestricted growth of any cell or body, is programming the death of all cells. After some time, the cells are killed by our body itself, and that's why we are having natural killer cells. So when any cell is infected by virus, normally they represent some proteins on their surface. They are called MHC. MHC proteins, major histocompatibility complex. There are two types of MHC, MHC1 and MHC2. MHC1 is present on all the cells, while MHC2 is present only on few cells. They are which are related with the immune response. And MHC1 are responsible for the organ rejection. You all might have heard that after organ transplant, the recipient has to be given with the immunosuppressants. Okay, the reason is to prevent organ rejection, because anything which is from outside, that is called xenobiotic, and our body will try to fight it, to reject it. One of the reasons for that is the MHC proteins, and this is the reason why for organ donation we prefer the siblings or the person who are having blood relation with the recipient, because there are less chances of rejection. They are having same kind of genetic structure, and that's why same kind of MHC protein. MHC2 are particularly helpful in the viral infection. They will rise a flare-like structure on the surface. So the natural killer cells can easily identify those cells, and they inject the lysins. That is the proteins which can kill that cell. So the body will destroy the virus infected cell totally. Along with the viral infected cell, the virus is also destroyed. So that is the mechanism of killing the virus infected cells. Any question in this, anyone? Now this is non-specific defense without identifying the organisms, and then comes in action if these two fail, or on first exposure normally this is sufficient. Sometimes the disease may show the clinical symptoms, sometimes it may not. So after these two, the third line defense or specific defense come into action. That is called third line defense. These two are part of natural immunity or innate immunity or constitutional immunity, that is the part of constitution of body, while this third line defense is called acquired immunity. This acquired immunity is gained after exposure to some organisms, and that is why it is called acquired immunity. So here the antibodies are produced by body. These antibodies are specific for a specific organism. That is, for Mycobacterium, a separate antibody will be produced. For Salmonella typhi, a separate antibody will be produced. For poliovirus, a separate antibody will be produced. So for each organism, a separate antibody will be produced by our body. Now, this immune response or the antibody production is triggered by antigen. And in response to antigen, our body produces antibody. This is called specific defense or acquired immunity. Now, this acquired immunity can be categorized in two categories. First thing is active immunity and second is passive immunity. Active immunity is the immunity in which our body actively participates in the antibody production. That's why it is called active. So, the antibody are produced by body by infected person himself. While in case of passive immunity, the person gets ready-made antibodies, which are produced by other person or anybody. In case of active immunity, this can be further categorized in two categories: natural or naturally acquired active immunity and artificial or artificially acquired active immunity. In both the cases, antigen enters the body and it triggers the antibody production. While in case of passive immunity, antibody is given. Directly antibody is given to the person. That is principal difference between active and passive immunity. Now, in case of active immunity, the naturally acquired active immunity, here the entry of antigen to the body is natural. Entry of antigen is natural. That is by clinical or subclinical infection. Some disease like chickenpox never repeat again. You will only acquire once in a lifetime. કોઈ એવું સાંભળ્યું છે કે લચકડા લાઈફમાં બીજી વખત થયા હોય? એનીવન? નહીં. કેમ નહીં થતા? Because once a person gets infected, the active immunity is triggered. Active immune response is triggered, and that produces antibodies. Along with that antibodies, the body also produces memory cells. Now these antibodies are produced by B lymphocytes. Okay. And when the antibody production starts, first step is the identification of the antigen. When the body identifies the antigen, it reaches the surface. After that, it goes to clonal selection. Normally, the B lymphocytes are having some surface receptors, and these surface receptors are capable of identifying the antigen. So when an antigen enters the body, it will be identified by the surface receptors of the B cells or B lymphocytes. Now, each B cell is having n number of receptors on their surface. Okay, so the production of antibody will depend on the binding site to which the antigen binds. Let me explain. Actually, the surface receptors are nothing but the peptides. That is the sequence of proteins, and the binding which takes place over here is nothing but due to either the hydrogen bonding or van der Waals forces or London forces. That is the big forces of attraction. Now, this hydrogen bonding occurs best when a highly electropositive and a highly electronegative species are near to each other. Agree? So, in proteins, we are having NH2, which is having hydrogens, and carbonyl groups or hydroxyl groups, which are having oxygen, or thiol group, which is having sulfur. So there is more probability of hydrogen bonding between these two, because these species are highly electronegative, hydrogens are highly electropositive, and there are chances of hydrogen bonding between them. Now, as we all know that hydrogen bonds are very weaker forces of attraction, weaker bonds. But if more than one bond is formed at a time, then the strength of the resultant molecule will be high. Clear? More than one bond is formed at a time, then the strength will be high. Agree? Okay. Suppose આ બેન્ચ છે એને મારે દોરામાં બાંધી ઉપાડવી છે, ઉપડી શકશે? સાદો દોરો નહીં ઉપડે, પણ હું એની આજુબાજુ 200 દોરા બાંધી નાખું પછી ઉપાડું તો ઉપડી જશે ને? એ જ થ્રેડ છે, પણ મેં 200 બાંધ્યા, that will lift the bench, કારણ કે 200 ની સ્ટ્રેન્થ ભેગી થઈ ગઈ. અહીંયા સેમ થશે. હાઇડ્રોજન બોન્ડ એટલો વીક છે, but if multiple hydrogen bonds are formed between the antigen molecule and antibody molecules, the bonding will be formed, and that's why the B cells will try to read more than one binding site. So, on the surface of an antigen and surface of an antibody, if more than one bonds are possible, then it will increase the strength of bond between them. It will increase the stability of the antigen antibody complex, and that is why the B cells will try to read those surfaces. Say, on the surface of antigen, if the structure is like this, NH2, COOH, NH2, the antibody which are produced by B cells will have complementary structure. That is, it will have NH, COOH, NH2 and COOH. So it can form multiple double bonds. Clear? And it depends on the amino acids, the configuration, their arrangement. That is why on first exposure, the B cells need to read the surface first. It is like a new recipe. If you are trying a new recipe at your home, you need to collect first all the information, all the ingredients, and you need to follow the steps or instructions. On second time, if you are preparing the same dish, you will be quicker. You can produce very quickly, because you now know what ingredients are required, what is the process, etc. etc. The same happens to the body. So on first exposure, sometimes the body may not be capable of utilizing that antigen, may not be capable of killing that antigen, and that's why we may experience the symptoms. That is called clinical infection. Sometimes the body is capable of killing the antigen on the first exposure itself, so we never experience any clinical symptoms, and that is called subclinical infection. In COVID also, you might have experienced that your family members suffered from COVID and you didn't. Agree? ઘણા બધાને એવું થયું હશે કે મારા ભાઈને કે મારા બહેનને કે મમ્મીને કે પપ્પાને કોરોના થયો, મને ના થયો. Because your body was capable to neutralize that antigen on first exposure. તમને કોરોના નથી થયો, તમારા ફેમિલીને થયો, છતાં પણ તમને ઇમ્યુનિટી મળી ગઈ, કારણ કે તમારો બોડી એના કોન્ટેક્ટમાં આવ્યું. And that's why you now are immune without even getting a vaccine. You were exposed to the COVID virus, but still you didn't show any symptom. You didn't reach to clinic, and that is called subclinical infection. If you don't see any symptoms, that is subclinical infection. So immunity can be gained even after subclinical infection. Again, so in first exposure, the surface reading takes place. The B cells prepare themselves to produce the antibodies. Now, the T cells also help them in this selection or in this binding. They are called helper T cells. The T cells are also two types: killer T cells and helper T cells. Okay. The both B cells and T cells are lymphocytes, and both are produced in bone marrow. But after production, these T cells migrate from the bone marrow to thymus, and they mature in thymus. When we get infected, the T cells are maturing in the thymus, while B cells remain in the bone marrow itself, they mature over their vessel. Initially, these B cells were isolated from the birds, and in birds they mature in the gland of bursa. That's why they are called B cells. In humans, they are maturing in the bone marrow itself, and that's why we call it B cells. While those cells which mature in thymus, they are called T cells. And this is the reason why during normal diagnosis also, when you go to a doctor, they will ask you, ગળામાં દુખ્યું છે કે અહીંયા કંઈ પેઇન થાય છે અથવા તો નાના બાળકોને તમે લઈ જાઓ then they will check ત્યાં ટચ કરીને જોશે તમે ખાલી તાવ માટે લઈ જાઓ તો એ ગળું ચેક કરશે. The reason is in case of infection there is swelling over here કારણ કે આપણી thymus gland અહીંયા છે એટલે અહીંયા swelling થઈ જશે કારણ કે T cell નું પ્રોડક્શન બોડી વધારે છે as a immune response. એટલા માટે અહીંયા ઇન્ફેક્શન થશે અહીંયા પેઇન થશે તમને પણ ઘણી વખત થશે તો ઇન્ફેક્શન લાગ્યું તો ગળું દુખશે બરોબર. Now, these B cells then go through clonal selection. Clonal selection, that is, these B cells are now divided in two different types. One, that is memory cells. They will remain as a backup, as a memory within the body. Now, some of these B cells are having a very long memory, a long lifespan. Sometimes lifelong. As I said for the chickenpox, the memory is lifelong. It will never repeat. કારણ કે એના બેકઅપ સેલ બોડીમાં પડ્યા છે જેવા ઓર્ગેનિઝમ ઇન્વેડ થશે the B cells will be triggered. They will produce the antibodies, utilize the antigen and will lead to the phagocytosis. So the memory remains lifelong. For some disease the memory is short-lived. For example, for tetanus, the memory is 10 years. And that's why we need to get vaccinated against tetanus every 10 years. બરોબર 10 વર્ષે tetanusની વેક્સિન લેવી પડે. Same is with the yellow fever. The memory is for 6 months only. For flu, the memory is for 1 year only. Every year we need to get a flu shot. Though in India we are not taking it, in US it is compulsory for all the senior citizens. Those people who are aged above 60, they are given flu shots in every September or October by the United States government. Because after October, November, the season of flu starts over. That's why they get vaccinated every year. So, a few of the B cells will multiply and they will remain as a memory cell, while the rest will act as a producer cell. They will produce the antibodies. Okay? They are producer cells. They will produce the antibodies. So, these producer cells are now circulating to the blood. They will produce the antibodies, they will target the antigen, they will utilize the antigen, the antibodies will utilize the antigen and will lead to phagocytosis. So this is called clonal selection. Two clones are produced and they are having two different functions. Now, the B cells which are produced over here, actually in our blood there are a number of circulating B cells. So, when one B cell is multiplying, it will produce the antibodies of a single type. Antibodies of single type. For example, this B cell has raised the surface of the antigen in this receptor site, at this receptor site. It will only produce the antibodies which are having the structure which is complementary to this site. Now, on a single antigen, it may have more than one binding site. Let me show these sites with a different sign. Okay, these are 4 different binding sites. In the enzymes, they need to locate the mechanisms. So for that representation only, I am showing this. So if the B cell reads this surface, it will produce the antibodies which are complementary to this structure. If it reads this surface, it will produce antibodies which are complementary to this structure. If it reads this surface, it will produce the antibodies antibodies which are complementary to this structure, and so on. Agreed? So, whatever surface of the antigen is read by the B cells, that kind of antibodies are produced by B cells. Now, in our blood, we are having n number of B cells, and each B cell reads a different surface of the antigen. જેના કોન્ટેક્ટમાં આવશે એ રીડ કરશે, and that is why different B cells will produce different types of antibodies, and hence our blood is a mixture of antibodies of different types. During COVID, you might have read about the plasma therapy. Okay, the person who recovered from COVID were becoming plasma donors, and they were donating the plasma to the patients who are infected, and they were getting good recovery. So the plasma is rich with the antibodies, but these antibodies which are present in plasma are polyclonal. They are produced from different clones. This is what clone, copy of one B cell. So they are produced from polyclones, multiple type of B cells. Now, if we isolate a single B cell, say for example this B cell only, all the copies of this B cell will have the same kind of, will produce the same kind of antibodies. Agree? Clear? All the copies of this B cell will produce a single type of antibody. So, if I isolate this B cells and I multiply it outside the body, the multiple copies of this B cells will be produced, and that B cells will produce further multiple copies of the antibodies or immunoglobulins, as they are produced from a single clone of the B cell. They are called monoclonal antibodies. Or MABs, MAB. The number of drugs, you might have read this word as a prefix, sorry, suffix, બરોબર? bosutinib, ઘણી બધી બરોબર મેબ આવે છે પાછળ. All these are monoclonal antibodies. They are produced by single cell. Now, while producing these monoclonal antibodies, as we know that B cells are having very slow multiplication or slow cloning capacity, and that is why they are fused with the cancer cells or carcinoma cells. And the fusion of this B cells with the carcinoma cell is called hybridoma. It's a hybrid. It is called hybridoma. So we are increasing the rate of multiplication of the B cells by fusing it with the cancer cell. That is called hybridoma, and that hybridoma will produce more number of antibodies quickly because they are multiplying fast. You can get multiple copies and at the same time you can get multiple antibodies. So this hybridoma technique is used for producing the monoclonal antibodies or MAPs. Clear? Any question is anyone? Now, the next part, that is artificially acquired active immunity. Here, the antigen enters the body artificially, not naturally. So, we are injecting an antigen which is either live or killed or attenuated, or we are only injecting a part of antigen, not entire antigen but a part of antigen. It may be a toxin produced by antigen which is neutralized, or it may be a cell wall component of antigen. Recently, we are also having RNA, messenger RNA, and DNA. So all these components can be injected, and that we call a vaccine. So the vaccination is for artificially triggered active immunity. We are triggering the active immunity artificially by injecting the antigen. Now, the vaccines are different types. vaccine is the suspension of microorganisms. Suspension of microorganisms, that is, the microbial cells are suspended. The cells may be lined. For an example, BCG vaccine. It is used for TB, and it may be killed or attenuated. For an example, TAB vaccine. It is used for typhoid and paratyphoid. It can be a toxoid. That is, the toxins are neutralized and they are injected to the person. Now, here we are injecting exotoxins which are neutralized by some treatment. The examples of toxoids are tetanus toxoid and diphtheria toxoid. These are two toxoids. Then they are having component vaccines. That is, only a part of microorganism is injected. For example, a cellular pertussis vaccine. A cellular pertussis vaccine. You might have heard of DTP. Okay? DTP vaccine. It is providing protection against diphtheria, tetanus, and pertussis. Diphtheria, tetanus, and pertussis. Previously, the DTP vaccine was available. Now we are having DTAP. That is diphtheria, pertussis, and diphtheria, tetanus, and cellular pertussis. We are only injecting a component, only a cell wall component of the bacteria, not the entire bacteria. Okay? Mody pertussis is the organism which is responsible for pertussis. Mody pertussis. So we are only injecting a cell wall component which is antigenic, which can produce the antigen response, that is trigger the antibody production. That is a cellular vaccine without any cell, entire cell. The reason behind that is, number of times what happens that because of improper inactivation of the cells, it may induce the disease. Before I think 5 or 6 years in UP, we have reported the occurrence of polio after polio vaccines. Currently, we are free from polio. I think 3 or 4 years back, India became free from polio. We have not reported any cases of polio since I think last 5 years. But in UP, the cases were reported because of the vaccination. That indicates improper inactivation. And there are more concerns about the whole vaccines. And that is why now the scientists are working more on the vaccines which are only having the antigenic component, not the entire organism. And as a part of that, this DTP is now changed to DTAP. Okay. Then modern vaccines like mRNA vaccines or DNA vaccines are also available. Okay. Apart from that, the vaccines can also be categorized based on the number of antigens present. It can be called simple vaccine, which contains only one antigen. For example, BCG. Here, this is the correct question of the examiners when it comes to MCQs. BCG, it is containing only a single species, Bacillus of Calmette and Goring. This is the name of the scientist who discovered this species, Bacillus of Calmette and Goring. It contains only single antigen and it provides a protection against tuberculosis or TB. But it's a single antigen. Its name is always written as capital B, C and G. And that's why sometimes it is misunderstood as a mixed vaccine. So it's a simple vaccine, only one antigen. When more than one antigen is present, it is called mixed vaccine or polyvalent vaccine. Polyvalent, a mixed vaccine, more than one antigen. The examples of such kind of vaccines are TAB. It contains Salmonella typhi and Salmonella paratyphi. A and B, two types of salmonella. There are typing and only one strain of salmonella typing. Then MMR provides protection against measles, mumps, and rubella. Then DTP, diphtheria, tetanus, and pertussis. Then currently we are having pentavalent vaccine in market. Pentavalent. It contains 5 different antigens, 3 which are listed under DTP. Apart from that, there are 2 more. One is HIV and second is HP. This is Haemophilus influenza B. Haemophilus influenza B and HP, that is hepatitis B. Jerry promotion age hepatitis B. That 5 different antigens are present. Recently, hexavalent vaccine is also included in the market. This is Penta. Hexa contains all these 5 components plus injectable polio, IPV, or injectable polio virus. So it contains 6 different antigens. Now, the logic behind combining more number of antigens in a single vaccine is to provide minimum shots to the kids, to the infants. In the world, there are a number of campaigns going on which suggest that vaccination is a cruel practice. And once upon a time, one of the president of the United States was also advocating that the vaccination should be reduced, or there should not be compulsory vaccination. The reason is the vaccines are painful to the kids, but at the same time they protect the kids as well. And actually vaccination is the only mean through which we can totally eradicate a disease. Mind you, it's the only tool through which we can totally eradicate a disease. You might have read or seen that your parents are having a coin like scar on their shoulders. So, રૂપિયાના સિક્કા જેવું મોટું સ્કાર હશે તમારા પેરેન્ટ્સને લગભગ બધાના. પછી પૂછ્યું છે શેનું છે? શેનું છે? હા, વેક્સિન. કઈ વેક્સિનનું? આપણા શોલ્ડરમાં કેમ નથી? એટલું મોટું ના હોય. આપણે નાનું જશે ખાલી એક મોતીના દાણા જેટલું, હે ને? એ લોકોને એક રૂપિયાના કોઈન જેટલું. કેમ? શેનાથી? So that is the આપણે ગુજરાતમાં જેને શીતળાની રસી, બરોબર છે? હવે એ ડિઝીઝ છે ટોટલી ઇરેડિકેટ થઈ ગઈ છે, વર્લ્ડમાં ક્યાંય જોવા નથી મળતું. That is possible because of vaccination only. તમે એન્ટીબાયોટિક્સથી ડિઝીઝને ટ્રીટ કરી શકશો, ક્યોર કરી શકશો, પણ એને ટોટલી નાબૂદ નહીં કરી શકો. While the vaccination is only tool to eradicate a disease totally. વર્લ્ડમાંથી સામે નાબૂદ કરી શકશો. પોલિયો જેમ ઇન્ડિયામાંથી સામે નાબૂદ થઈ ગયું, that is because of the vaccination. જો આપણી પોતાની ડિફેન્સ સિસ્ટમ એટલી પાવરફુલ થઈ જશે, then કોઈના બોડીમાં ઓર્ગેનિઝમ્સ મલ્ટીપ્લાય થશે જ નહીં. મલ્ટીપ્લાય નહીં થાય તો સ્પ્રેડ નહીં થાય. સ્પ્રેડ નહીં થાય તો ડિઝીઝ ક્યાં જવા સ્પ્રેડ થઈ નહીં શકે. જેવા તમે કોન્ટેક્ટમાં આવશો તમે તે ઓર્ગેનિઝમને કિલ કરી નાખશો, બરોબર? નાઉ ફાઇન્ડ આઉટ, vaccination is not only helping you but also helping others indirectly. One of the word you might have heard is called herd immunity. Our word COVID માં બહુ ફેમસ થયો, herd immunity. Okay. Now, technically the meaning of herd immunity is herd એટલે ટોળું. બરાબર છે? So herd એટલે ટોળું, બરોબર છે? કમ્ફર્ટેબલ છે ને બધા? any problem anyone? So the meaning of herd is a group of people or group of animals, whatever. For that we are using word. Now herd immunity is important. Let me give an example that sometimes some kids are immunocompromised. This immunocompromise, they are immunocompromised because of some disease or some treatment. So suppose a child is suffering from cancer and the child is going through the treatment of anti-cancer drugs. Now it is a normal side effect of anti-cancer drug that they are suppressing the immunity. They are immunosuppressants. If such kid come in contact with any infectious organism, then the kid is more prone to infection. Agree? Okay. Now imagine a situation where, and at the same time, mind well, at the same time, we cannot give vaccine to such kids. બરોબર છે? જેની ઇમ્યુનિટી ઓછી છે ને એને વેક્સિન પણ ના આપી શકે, કારણ કે in that people the vaccines can produce severe infection or severe response. બરોબર? એકદમ અનકન્ટ્રોલ્ડ રિસ્પોન્સ પ્રોડ્યુસ થાય. આપણું બોડી છે એ એન્ટીજન ન્યુટ્રલાઇઝ કરી નાખશે એટલે વાંધો નહીં આવે. જે ઇમ્યુનો કોમ્પ્રોમાઇઝ છે એનામાં વધારે સિવિયર રિએક્શન થશે. That's why they cannot be also vaccinated. So to protect those kind of people, if assume that out of total class of 100 students, 90 are vaccinated or 99 are vaccinated, only one is not. Mind it. And that person is always playing with them, always roaming with them. Now, if any of those 99 students get infected and if they are not vaccinated, for example, they will transmit the disease to that unvaccinated person, the immunocompromised person. But if the 99 are vaccinated, even though they get the infection, they will not carry that infection, they will not spread the infection because their body is capable of neutralizing that antigen. એને disease નહીં થાય. disease નહીં થાય એટલે ટ્રાન્સમિટ નહીં કરે. ટ્રાન્સમિટ નહીં કરે એટલે તમે વિચારો કે 100 જણામાંથી એકને જેને વેક્સિન નથી અપાઈ એના સુધી disease પહોંચશે જ નહીં. And that person is protected. So the vaccination is also helpful to others as well. If you are not transmitting, you will be protected. એટલા માટે જ ખાસ ગવર્મેન્ટે સૌથી પહેલા કોરોનાની વેક્સિન કોને આપી હતી? ફર્સ્ટ લાઈન વોરિયર્સ. જે લોકો કોવિડના પેશન્ટને હેન્ડલ કરે છે એને પહેલા આપી. કેમ? તો કે એ જો ડિસીઝ ઘરે લઈ જશે તો એના ઘરે પણ થશે બધા. એટલે સૌથી પહેલા એને આપી દીધી. એ આખો દિવસ કોવિડના પેશન્ટની સાથે રહેશે પણ એને કોવિડ નહીં થાય. નહીં થાય એટલે એના ઘરે નહીં રહે. અને ઘરે નહીં રહે એના ફેમિલી પણ નહીં થાય. બરોબર? So that is called herd immunity. So for that also જે પણ કદાચ ફાર્માસિસ્ટ તરીકે જોબ કરતા હોય definitely you are working with vaccines. It is a duty to spread the awareness about vaccines. Now next part is the passive immunity. Most time કેટલા વાગે છે? 12:20. બની જાઓ 10 minutes more. over 10 minutes માં જેટલું થાય એટલું આપણે મેનેજ કરીએ. Now the passive immunity, as I said here, the ready-made antibodies are given. This passive immunity can also be categorized into two categories: natural and artificial. Natural and artificial. Now, how can an antibody synthesized in other person can enter to a body without any artificial means? Yeah, child. Okay, mother to child, mother to fetus. Okay, these are only two ways. First thing is the immunoglobulin G or IgG molecules. They are having the capacity to cross the placenta. Okay, and by crossing the placenta, they can enter the body of the infant, that is the fetus, and thereby it provides the protection. And this is the reason why some disease like chickenpox will not affect a kid up to 6 months, infant up to 6 months. 6 મહિનાનું થાય ને બાળક ત્યાં સુધી નસતા નહીં કરે, કોઈ પણ કન્ડિશનમાં નહીં કરે, કારણ કે એના મધરના બ્લડમાંથી એને એન્ટીબોડીઝ મળેલા છે. બરોબર છે? So this is first, and second is the colostrum. colostrum, that is a yellow color fluid which is present in the breast milk. It is secreted along with the breast milk. It is rich with the IgA, immunoglobulin A. And that is why during breastfeeding, the mother imparts these IgA molecules to the kid. And that also provides protection against number of diseases. So these are two means by which the antibodies can transfer naturally from mother to fetus and from mother to breastfeeding kid. Clear? So this is natural transfer, and artificial is by providing the serum or anti-serum. Serum or anti-serum. COVID માં જે પ્લાઝમા ટ્રાન્સફ્યુઝન કરતા હતા, that is also a part of artificial passive immunity. ત્યાં પણ આપણે એન્ટીબોડીઝ આપીએ છીએ, બરોબર? Sometimes these antibodies are synthesized in other animals or humans, and they are injected to the person, that is infected person. Clear? So with this we are completing finishing today's lecture. If you have any question, you can ask. Anyone? હજી ઘણું બધું બાકી રહી ગયું છે. If time permits, we will discuss later on. If you have any question, you can ask.
નોર્મલ ઇન્જેક્શન આપીએ ત્યારે કેમ માર્ક નથી?
નોર્મલ ઇન્જેક્શનમાં
કેમ એવું માર્ક નથી થતું?
કેમ માર્ક નથી થતું? ઓકે, નોર્મલ ઇન્જેક્શન આપીએ છીએ ત્યારે માર્ક એટલા માટે નથી થતું કે નોર્મલ ઇન્જેક્શન્સ જે આપીએ છીએ ને નોર્મલી નિધન નાની હોય છે. બીજી વસ્તુ, વેક્સિન્સ આપીએ ને ત્યારે એને સબક્યુટેનિયસલી આપે છે, બરોબર છે? અને સ્કિનનો જે પાર્ટમાં આપે છે ને એ ભાગ સ્ટ્રેચ રહેશે, બરોબર છે? એટલે ઘણી વખતે થાય છે ને નાની નિધનથી આપેલા છે તેનો માર્ક નહીં રહે. કોઈના શોલ્ડર ઉપર કોવિડ વેક્સિનનું માર્ક નહીં રહે, કારણ કે કોવિડ વેક્સિનનો ડોઝ નાનો છે અને એકદમ પાતળી વેક્સિનથી આપેલું છે. એવી જ વેક્સિનના માર્ક રહેશે કે જે જાડી નિડલથી આપેલું છે. ખાસ કરીને ટેટનસ, ધનુરની રસી છે ને એનું માર્ક બધાના બોડી પર હશે, કારણ કે ધનુરની રસી જે આપવામાં આવે છે ને એની નિડલ થોડી મોટી હોય છે. Any other question, anyone? No more questions? Let's finish then. Okay, thank you.